Okay, yeah, it looks like the participants are joining. Welcome, everyone. I'm Steve Pipe from the University of Michigan, and welcome to this live webinar from the ISTH called Ask Me Anything. We're going to be addressing the current status and the future of gene therapy and hemophilia in North America. Purpose of this is to provide some expert perspectives on the current and future state of hemophilia gene therapy, lessons from clinical implementation, some emerging innovations, and evolving treatment paradigms in North America. And this educational activity is supported by independent educational grants from BioMarin, CSL Behring, and Pfizer. And joining me today is Dr. David Lillicrap.
Steve, good morning. Good morning, everyone. Good to see you, Steve. Looking forward to an interesting hour of dialogue about where we are with gene therapy for hemophilia.
Yeah,
I guess I should just say I'm in Canada, so this is truly a North American, although my co-presenter this morning is actually Canadian, as many of you know, now in the US. I'm at Queen's University and have been interested in gene therapy for hemophilia now for at least a couple of decades.
Yeah. And I think between the two of us, we have a nice complement of a long track record of basic science investigation in gene therapy and hemophilia, as well as clinical care and clinical implementation of gene therapy. So to kick us off, David, we're going to start just sort of talking about the state of the field. So how would you describe the current state of hemophilia gene therapy in North America, and what has changed most dramatically over the past few years?
Yeah, so I think we should, first of all, remember that clinical gene therapy successes now are 15 years plus old. So the original clinical study that was done by Nathwani and the group at St. Jude was published in 2011. And those patients are still going with factor 9 gene therapy levels of factor 9 between about 3 and 5%. And so we've got a reasonable history now of successes with factor 9 gene therapy. I think the other lessons we've learned along the way is that this is very complicated. As you know well, basic and clinical studies now have been ongoing for a long time, and we still haven't hit the sweet spot. Things are much better and I think still exciting. I'm really encouraged about the innovation that continues. But I think that the current clinical application of gene therapy, especially for hemophilia A, continues to be a challenge and reminds us that this type of therapy is difficult. It's challenging. It has wonderful advantages when it works well, but it's not an easy accomplishment. So maybe I'll stop there and get your input.
Well, if I think back maybe even 3 years ago, you could put up a table of multiple clinical trial programs active in hemophilia A and hemophilia B. And yet now in North America, if you look at the available gene therapies for hemophilia, we have a single approved and continued marketed product for hemophilia B. That's Hemgenix from CSL Behring. So what's happened?
Yeah, so really these are lessons along the way that I think maybe we anticipated to some extent. But honestly, I've also been a bit surprised. Even the Hemgenix product, which is now licensed and available in Canada and certainly in the US for now well over a year or so, there were current issues about availability, or there were issues of availability 2 or 3 months ago because of production or issues around the stability of the product. So again, it reminded us that even after an extended history of the development of gene therapy and of clinical trials that were ongoing for several years, the introduction and regular clinical use of these agents is not straightforward, which is in a way a little surprising, but it just reminds us that this is complex.
Yeah. I mean, we can reflect a little bit on the launch of Roctavian in the US. This was the first out of the gate with a hemophilia A gene therapy. This AAV5, similar to what's used in the CSL Behring product. The data for the BioMarin product is now, we have data all the way out past 5 years, and even longer if you include the phase 2 program. And if you look at the 5-year data that was summarized just within, the last 6 months or so, roughly 70% of the patients remain off of prophylaxis and with factor levels that are in the mild hemophilia range, at least mild to non-hemophilic range. That does leave about 30% of patients who've either returned to prophylaxis or still have levels that are below maybe what the targets we anticipated. And I think there's a proportion of patients who actually have less than 3% activity by a chromogenic substrate assay, but have chosen not to resume prophylaxis because they're not bleeding. And, there were still patients who were seeking this platform of therapy. However, as of the end of last month, Roctavian has been withdrawn from the market as well. And so maybe we can talk about what were we anticipating we should be seeing from a gene therapy product for hemophilia, and what are we sort of reaching the limits of being able to deliver, particularly when it's being compared to other new innovative therapies that are coming to the marketplace?
Yeah, so let me answer sort of in reverse and go to the current menu for hemophilia therapies, which, as we all know now, is incredible. There's a wonderful diversity of possibilities for therapy of both hemophilia A and B, and actually particularly for hemophilia A with the factor VIII mimetic agents. So for a couple of decades, people like you and I were standing at podia saying, molecular biology is going to solve hemophilia. And it didn't happen, and it didn't happen. And then all of a sudden, it seemed, over a few relatively short years, we now have the fruition of the basic science that was initiated in the mid-80s. So I think that the landscape is very challenging for hemophilia. And especially, as we well know, the differentiation between 8 and 9 is very important. You and I know from the basic science studies that we've been involved in over a couple of decades that factor VIII is always challenging. I love to refer to it as the prima donna of hemostasis. It really challenges us even when we're doing so well with factor IX. And of course, the factor IX advances have in part been made available because of the 9 Padua gain of function variant, which has boosted the levels of activity of factor IX 8-fold. So that's wonderful. But so far, we haven't found a similar construct yet for factor VIII, although that's being worked on. And there are some possibilities that are in very early phase preclinical assessment. So I think when we talk about hemophilia gene therapy, it is important to keep that differentiation in mind, because the Nathwani group are now out to 14, 15 years of consistent expression. Honestly, there's no evidence that factor IX levels are falling in those patients. So it could well be that individuals who've received factor IX gene therapy may have persistent levels of therapeutically beneficial factor IX for a long period of time, which is wonderful. That's a great outcome. But the factor VIII study is, I think, the interaction between industry expectations and the clinical data are not simple to put together. I'm not sure I even now understand what happened there. Maybe our expectations were just too high, and for sure, the results were somewhat disappointing. But as you pointed out very well, there are still patients who are doing well and are not back on prophylaxis because their factor VIII levels out at 7+ years are doing okay. Tell me, what do you think we've learned from the real world application of gene therapy, how has that been different to what we saw in the previous clinical studies?
Yeah, it's pretty interesting. Having been involved in clinical trials for many years with gene therapy, and then shifting to clinical implementation. Some of the things that are really different about those two settings are not insignificant. Clinical trials are fully funded. The research and the medical teams are compensated. All your institutional administrative costs are paid. You have investigational pharmacies who handle the procurement and processing. Often patients have their travel and expenses reimbursed, and we're doing all of this in a very controlled environment. But then when we switch to doing commercial gene therapy in the clinical setting, now we have to find a way to sort of incorporate that into our, if you like, day-to-day practice. That it means having a broad multidisciplinary team who are doing education with the patients and the families, helping them understand what gene therapy could offer. Trying to identify patients who are most suitable and could really benefit from what gene therapy can deliver. And then we have to have a plan for how we're going to implement that at our site. We're going to be shifting to the clinical pharmacies from the research pharmacies. We are having to now incorporate infusions, often in a very busy infusion center that's already doing all kinds of other standard infusions. We have to bring them up to speed on doing gene therapy infusions. What I've appreciated the most from gene therapy as it's currently applied for hemophilia, a single infusion over several hours, relatively infrequent and manageable infusion-related reactions, and essentially entirely an outpatient managed procedure. This is very doable within the hemophilia treatment centers that we have in the US and in Canada. We have very strong, highly specialized multidisciplinary teams. And as long as you bring everyone along on that learning pathway, and then you do your groundwork to get your implementation ready, we've shown that we can actually deliver this within our HTCs. Now, in some cases, I know some of my colleagues have actually merged with their existing gene therapy and cellular therapy programs, and that is another alternative. But in the U.S., we have 140 centers across the U.S., almost every geography. Not everyone's going to have those kinds of resources. So we've tried to do a very good educational process to help everybody come up to speed. And I think what I've appreciated from clinical implementation of gene therapy is I believe any center can actually do this, and any patient has an opportunity to benefit from this therapy. And so it's really, I think, been a success story of taking what you identified as a really complicated therapeutic, but make it accessible for patients in any geography and be able to get this therapy and benefit from it.
So, Steve, as you know, in Europe, the hub and spoke model is something that they think is very important. But I think the geographies in Europe compared to North America are very different, right? I mean, sometimes we have patients who come 3 or 4 hours to see us in the hemophilia centers. In Canada, where, by the way, we had our first Hemgenix infusion now 3 weeks ago, so it's happening. I think there are likely to be no more than 6 or 7 centers that do hemophilia gene therapy across the country, which means that certainly some patients will need to travel quite a distance to get their infusions, and they will return, presumably at some point, to a more peripheral center. And so there will be a kind of a pseudo hub and spoke. Whereas in the US, I think, as you pointed out, eventually there may be 100 centers who are capable and will do gene therapy. Do you think that will happen?
Yeah. Well, I think if the demand was there, that may happen. I think what's happened now is certainly centers that had no experience doing investigational gene therapy were able to deliver commercial gene therapy. So that happened. And sometimes in fairly remote portions of the country, they were able to execute this. But at the same time, we also have seen a hub and spoke type mechanism. It started in the clinical trials where patients sometimes went to other centers for their dosing and then came back to their home center for the rest of their follow-up. I actually did that with one of our Roctavian patients. We did it jointly with a partner center, and they did the infusion, and then the patient came back to us for the follow up. So I believe that can happen and it can work well. I think ideally, though, patients have a long history at their centers. They have had deep involvement with the multidisciplinary team and particularly their physicians. And they would like them to carry them through this therapy as well. And I think no matter how you do the mechanics of the infusion, I think all the centers need to be capable of doing the follow-up and then also dealing with the long-term data collection, which I think is going to be critical for decades to come for all the patients who have received gene therapy.
Right. I want to pick up for a second on that last comment you made about the long-term follow-up, and maybe actually return to the Roctavian studies for a moment. So their large phase 3 study with 132 patients, there must be now probably in the region of 200 patients who've received Roctavian in the clinical trials and then subsequently in commercial clinical delivery. It would be absolutely critical to try to follow those patients beyond 5, 10, 15 years. And I think we're all concerned how that's going to happen. There are registries, as we know. The World Federation has a registry, and I think there are individual country registries. I think you have an Athens registry. We have a Canadian bleeding disease registry as well. So following any of these patients who received any of the gene therapies long term is extremely important. It's important for research issues, but it's also very important for safety concerns. There is still these potential long-term genotoxicity concerns, which we will only be able to address by following patients for 5, 10, 15 plus years. So hopefully the people who've received Roctavian, because the product has now been withdrawn, that there will be measures in place to make sure that there's adequate follow-up. Do you think
I think the genotoxicity issues, I think we're all pleased that of what we've seen and what we know from the data from the trials and the long-term follow-up to date. I think if we look to other genetic interventions over the years, when we've seen true evidence of genotoxicity, it's actually happened fairly early. And so I think the types of genotoxicities that maybe we're talking about now are maybe more nuanced, epigenetic changes, maybe, two factors interacting together, underlying liver inflammation on top of a genetic modulation, et cetera. And those may take, some considerable amount of time to manifest themselves. So in some ways, I think we've learned a lot about the safety of this particular platform of therapy, but it's absolutely critical, as you said, that we continue over not just a few years, but decades. And I know the companies probably have their own regulatory requirements, even if they're no longer marketing their products, and hopefully that will continue. But ultimately, I think it does fall on the individual centers, and whatever data systems we're using, we have an obligation to provide that data. And it's not going to just inform this platform of therapy. I think it's going to inform future innovations, and it's also going to inform other diseases. There are lots of other gene therapies that follow a similar mechanism of an AAV delivered to the liver. And the observations that we have on safety and efficacy also have implications across other disease states. I was wondering if we could talk a little bit about what you see as the biggest scientific barriers that still need to be overcome in order to make gene therapy ultimately as effective, durable, and broadly applicable as what I think people are looking for.
I think the biggest step forward would be to find other ways of delivering transgenes. I think that viral vector delivery has proven to be effective and safe, but I think that there are still better ways of doing this, and maybe even safer ways of doing this, and certainly more targeted ways of doing this. And so I think that the next steps in improvement of hemophilia gene therapy, which likely will involve probably some form of genomic editing type of process, also needs to incorporate that in a delivery system, which may not involve viral vectors. Now, the problem, as we all know, you and I certainly know, is that, again, the development of things like lipid nanoparticles is very complicated. There are many groups now trying to work with this. The toxicities are relatively minor, but there are other issues which need to be overcome. So I think the delivery system is really important. And then for factor VIII, I think that nature has told us that the cells that like to make this are a very unusual subset of endothelial cells, and that unless we do something to help liver cells, expressing transgenic factor VIII in liver cells is going to be problematic. And I think, again, many of us believe, and you've done studies in the past that say that as that protein traverses the endoplasmic reticulum, the folding that occurs is critical. And if that isn't controlled, you end up getting cellular stress, which eventually leads to cell death. And probably what was happening with Roctavian, it is in the first 2 or 3 years, the cellular stress was sufficient to reduce the amount of factor VIII that was expressed until a comfortable level of factor VIII was reached, which is about 15 to 20%. So again, I think there's 9 and 8 considerations which are different, but I think delivery systems are going to be critical in the future.
Yeah, you highlighted a key issue. Any way you look at the success of hemophilia B gene therapy as currently applied, you have to keep coming back to the factor IX padua. The ability to boost the activity by 6 to 8-fold with that single point mutation without changing dramatically the structure of the transgene really was a key innovation and really made, I think, gene therapy a reality for many Hem B gene therapy recipients around the world. For Hem A, we've been missing the right Padua-like variant. But there's been some recent innovations, designer approaches to factor VIII that I think offering some promise. You mentioned that sort of comfortable level of factor VIII expression. This is actually the approach that Roche and Spark Together are taking with their program, because they had a B-domain deleted version of factor VIII, which was giving what seemed to be durable expression in that, teens level of factor VIII expression. And they've presented their long-term data with that initial construct. And they're now investigating point mutations that render some activated protein C resistance to factor VIII that probably is going to have some clinical benefit on how the factor VIII performs in bleed control. And so that's being actively investigated in a clinical trial. So that's one of the ongoing clinical trial programs that I think people are still waiting to see. There have been some new published naturally occurring factor VIII variants. The factor VIII Aurora, which was described in Colorado, is a single point mutation, which seems to substantially boost the activity of factor VIII. And that's another possible Padua type variant that maybe could be considered in future gene therapy innovations. So I think we still may, with the current platform of therapy, see a traditional AAV gene addition system for hemophilia A that might prove to have the type of long-term durability and expression levels that I think our patients want to see.
I totally agree. I think these developments are exciting. There are several variants that are being looked at. One of the things we should keep in mind is the immunogenicity of factor VIII. I mean, that's almost going to be historical in some people's minds, actually. But with the factor VIII mimetics, who worries about factor VIII immunogenicity? So every time you start changing coding sequences, we need to just be a little cautious about immune responses. To my knowledge, I think there's only been one patient who's received hemophilia A gene therapy who developed an inhibitor. Um, but and there's actually reasonable evidence that hemophilia gene therapy might be a tolerizing way of actually getting over inhibitors. BioMarin had that small study, which I think involved about 5 or 6 patients, which was prematurely stopped, but it looked somewhat encouraging. So it's too bad in a way that we're not going to be able to address that possibility of gene therapy, factor VIII gene therapy, as a way of tolerizing individuals who are either at risk or have a factor VIII inhibitor. So so I agree that transgene alterations would be very good, and there are some that are going to be looked at. Realistically, we won't know from those clinical trials for at least 2 or 3 years, I guess. Would you say? Yeah, it's going to be a little while.
Yeah, for sure.
Yeah. So in terms of the next generations of gene therapy, so we've talked about the transgene changes. What else do you see? So actually, maybe can I just return for a second to the one licensed and commercially available gene therapy, Hemgenix, has this peculiarity that we know about. Of course, you can give this to people who have pre-existing antibodies for Hemgenix. What do you make of that? Can you understand that? And is there any evidence that that is a concern at all? Because I think I've seen a little bit of data suggesting that there are some infusion reactions that occur with individuals who are still eligible for Hemgenix. The titers are not so high, but during the infusions they run into maybe minor infusion reactions, which we should keep in mind. So what do you make of that phenomenon?
It certainly rails against what was considered dogma that pre-existing immunity against AAV was a non-starter for getting an efficacious expression. AAV5, not naturally circulating in humans. Probably the antibodies that we're detecting in these assays are cross-reactivity from exposure to other AAVs. It could be the binding affinity of those antibodies is less so that even though these are giving a readout of blocking transduction in an in vitro assay, maybe in vivo when you're giving, billions of particles to a patient, that you can override the effect of those antibodies, and we're still able to deliver enough, enough gene transduction. I think the fact that Hem B is maybe an easier target for the reasons we talked about, and that even if you are dampening down the transduction efficiency by the presence of those antibodies, you can still get enough factor IX expression that's clinically relevant for patients. So I think it's probably unique to that particular capsid and the application in Hem B, because as we've already talked, AAV5 is also what's used in Roctavian, and I don't think we have any evidence that you can treat through pre-existing antibodies with that construct. So it is a benefit for patients because it's broadened the eligibility for who can actually receive this. I think we're still not there to know whether you can redose patients. The levels that are achieved, the antibody levels that are achieved after you've been exposed to gene therapy once are just seemingly insurmountable. And we don't actually even know if there is a cutoff where it's inevitable that you would not see expression. We did have one single patient in the phase 3 program who had a what would be considered a very high naturally occurring antibody level of titer of over 3,200, and he was a non-responder. But we don't know exactly where the cutoff. In the trial program, up to just under a 1 to 700, we still had good expression in patients. So there's an ongoing trial that's looking at that interval between 700 up to whatever they can get in the clinical trial program to try and identify where those cutoffs should be.
Yeah, I must say I'm not optimistic about redosing because I think the numbers of viral particles we give are so huge. I think, as you know, when we followed our dog model for 12 years, at 12 years, they still had very high levels of antibody, which would have prevented redosing. So I think that's going to be a major challenge. There is something in the.
If I can interrupt, we have two questions in the chat.
Yeah. What?
Very good.
How can we define the optimal patient who will best benefit from gene therapy besides indications, contraindications? How can we find the patients with best treatment response or establishment of criteria possible?
Well, I mean, I think that's a relevant question for me. Even though I'm doing gene therapy, clinical trials, et cetera, I am a pediatric hematologist, and honestly, I think we're still doing gene therapy too late. I mean, the clinical indications are still for age 18 and up, but when I think about the potential benefit of what I've seen in patients who've received gene therapy, this would be such an awesome experience for a younger adolescent patient to be able to live with what many have identified as a hemophilia-free mindset through those critical years in later school age and through adolescence. And you think about all the key decisions that a patient's going to make in their life about vocation they choose, where they're going to live, who they're going to marry, what sort of lifestyle they want to live. To be able to make those decisions without having to be thinking about your hemophilia anymore. I've seen it firsthand in patients who've received these therapies, including Roctavian, including Hemgenix. And so I really want to see us being able to deliver that to even younger patients. And that's probably going to take some additional innovation. But maybe getting to closer to the question here is, once you have the indications or contraindications dealt with, can you identify things that really identify gene therapy as what's best for a patient? And I think this is something that we talk about at the multidisciplinary team level. Who really just wants to be done with regular prophylaxis? Who wants to live their life without having to think about hemophilia anymore? And we can identify those patients in some short order when we go through our patient list. And there are some patients who've come to me sometimes with somewhat surprising reasons why they want to choose gene therapy. And maybe this is more applicable to the US experience, David, but there's some patients who are very concerned about the cost of their hemophilia therapeutics. And they see gene therapy as a way to sort of remove that anxiety off the table for the future. If they could get converted to even a mild phenotype or even into the normal range, then they don't have to worry about, is my insurance going to cover the types of therapies that I want to be on in the future? And so that's a real driver. As far as identifying a patient who's going to be in the upper bounds of the expression, after receiving this therapy, who's going to avoid some of the complications like infusion-related reactions, transaminitis related to, immune reaction to the vector. Unfortunately, I don't think we have identified those clinical factors, not that you can identify before the patient receives those therapies. When you look at the success of the hemophilia B program from Hemgenix, there's no question that the patients who were free of liver reaction, free of having transaminitis, had the higher overall expression. And those who did have an immune reaction to the vector, they had substantially lower expression overall. So if we could only identify who's going to or not going to have that reaction, that would certainly maybe influence who we select for that therapy. So right now, I think we're more focusing on the psychosocial aspect of who would benefit and trying to identify those patients first.
Yeah, so totally agree with Steve. I think in this age of personalized medicine, each individual person in total, I think saying that this is a perfect person for gene therapy is kind of, it will happen. But I think it's a complicated issue of identifying those people. I think in terms of predictability, one of the problems with factor VIII gene therapy for sure was that if someone were to say, what is my factor VIII level likely to be 6 months, a year, 2 years after I receive this therapy, it would be very difficult to do that. The predictability values for factor VIII gene therapy were not good. With Hemgenix, it's not entirely predictable, but it is a lot better than with factor VIII. And I think that you could say not only are you likely to get a level that is somewhere around, I mean, the means and medians have been about 35-40% of activity, but the durability, as shown by the original Nathwani study and more recently the longer term Hemgenix data out to what now 6 years, almost 7 years, looks very, very encouraging. So I think that we will be able to give confidence to patients who are interested in gene therapy. In terms of the ideal patient, that they have to be willing to be engaged with the clinic, the place where the treatment was given, or at least an active center, for at least 6 months, because the first 6 months is when these liver reactions tend to occur, if they're going to occur, and they require fairly intensive investigation and management. So I think even some of the older patients, Steve was talking about pediatric patients, I understand his frustration and say, we need to be treating children, that for sure we do. But if someone were to come to me and they were in their 60s, I think that there's no reason to believe, as long as their liver health is reasonable and there are no other significant comorbidities, why I would suggest they should not be considered for gene therapy. So I think at both extremes, we've got possibilities. Steve, there's some more things in the chat, I think. Yeah.
Yeah, I think you should address this one, David. This is from our colleague, Prasad Matthew. He said, I understand the reports of malignancy during the trials, assessed as not related to gene therapy itself. But recently, you're aware of the New England Journal paper that showed this case report of an AAV gene therapy recipient for a different condition. I think it's a mucopolysaccharidosis. It developed a neuroepithelial tumor, and this was in a child. So what do you think is the implication of this for hemophilia patients?
Yeah, thanks. It's a really important question. And I think what we can say is that the hemophilia cases who've developed cancer, those patients who've received hemophilia gene therapy and have developed cancer, there's about 4 of them, maybe actually 5 or 6 now, but they've all undergone extensive examination of the tumor material to see whether there's any involvement of the vector sequences. And in those instances, there's been no evidence of vector involvement in the tumors. Now, one should say that investigation is very complicated. It currently can only be done really in sort of 2 or 3 places in the world, and it's expensive. So every time we say, send this tumor, this cancer for investigation, this patient received hemophilia gene therapy 3 years ago, we want to know whether it was involved in the cancer. That's an important and difficult decision. It's going to take time and money. There've now been several thousands of patients who've received AAV gene therapy for different conditions. Many of those patients are young children who've received AAV gene therapy for SMA. And so that's treatments which have occurred in the first few years of life. And all those follow-ups have been relatively small. So I would think, within 5 or 6 years. So we haven't seen an incidence of cancer in those patients at all, but it is early days. And remember, I say many of these patients are very young children. The report of the recent neuroepithelial tumor was in a young child with Hurler's syndrome, a storage disease. And the things there to keep in mind are, A, they had a cellular pathology, which was very significant, which hemophilia patients don't have. This was a very young child who received extremely high doses of AAV9 vector, so a different serotype, directly into the central nervous system. And so, and also the transgene there was driven by a very strong regulatory element, promoter enhancer element. So there are a number of distinct features of the development of that tumor, which I think are distinct and very different to what we're doing with hemophilia gene therapy. So we need to be cautious. We need to follow these patients long term. There is no danger signal about oncogenicity at this point in time, but the patients must be followed for 10 and 15 years and, well, really for their lives.
Yeah.
There's a question about assays. I don't know if you want to take that. So this is the difference between the 1 and 2 stage responses, probably with both factor VIII and factor IX, actually. Do you have any comments?
Yeah, this is something I've been interested in for a long time. And, we've been dealing with assay discrepancies in the recombinant era when we moved away from human plasma. And in some cases, this is related to subtle differences in how the molecules act within the assays. Sometimes you can attribute this to post-translational modification differences between the molecules. We saw this exacerbated when we started to modify the constructs through bioengineering. So this came to light, particularly when we moved to B domain deleted forms of factor VIII, and there were discrepancies that were highlighted there. And this becomes interesting in the setting of expressing factor VIII in a non-native cell, like the hepatocyte, as you indicated earlier. And what we've also done with gene therapy is we've done what's called codon modification or codon optimization. And in that case, we're changing the codons that are used without changing the amino acid that it codes for. But it's interesting that the codons that are used in humans also direct the pacing of how a protein folds. And when you do codon optimization, you change that pacing of the protein extension and folding. And it's possible that this is introducing some subtle changes in the structure of the molecule, which we haven't been able to fully characterize. But it's been universal for factor IX and factor IX patois, at least, as well as with the B domain deleted constructs that are expressed in the hepatocyte. And there, the 1-stage assays seem to be about 1.5 to 1.6 fold higher than the chromogenic assays. Which one is the truth as far as the clinical activity is concerned? That's really hard to tease out. I don't think we have convincing evidence one way or the other of which one. For hemogenics, we use a 1-stage assay to read out the patois, and for Roctavian, we use a chromogenic assay to read out the factor VIII. But that difference has been observed for both. What is interesting, and this has been published in preclinical data, when you revert back to a wild type factor IX in the gene editing setting, so if we look at using CRISPR-Cas9 to direct factor IX expression in the hepatocyte, then that discrepancy seems to go away. And so when factor IX is then, as a wild type construct, is expressed within the hepatocyte, we no longer see that 1-stage, 2-stage discrepancy. So I think it's a nuance related to how we brought these constructs into gene therapy in the first place. And it's possible that these will be non-issues in the future as we shift to different innovations for how we do gene therapy going forward.
I think your comment about we still don't really know which is the true number, the chromogenic or one-stage numbers, and I think that's been a conundrum now for 20 or 30 years. So when we're just adding an extra level of complexity to that understanding with gene therapy. One of the other questions I think they'd like us to address is this issue about if we see patients now who are considering a change in their therapy, and they ask us, okay, well, it looks as though gene therapy is it's doing okay, actually more than okay for factor 9, is doing very well, but for factor 8, we're not sure. What do we tell them about what to expect in the next, let's say, 5 years? And to me, that would actually not be such an easy discussion, because the great thing is that there's still a ton of innovation happening. I mean, I think that we should all be really excited and happy that people have not given up on genetic therapies. I personally believe that in 10 and 15 years, there will be significant numbers of patients who will be treated with some form of genetic genomic therapy. But it will take a while to get there. So I think innovation will continue, and that's great in the long term. But in the next 5 years, I'm not entirely sure what's likely to be on the horizon and will be entering clinical application. What do you think?
Yeah, I think there's an important principle here that, when we talk to patients today about AAV gene addition, gene therapy in a commercial setting, we're building on a track record of learning that goes back 30 years. And you had mentioned with these current constructs in AAV delivery, we now have 15 plus years of clinical follow-up from clinical trials, and then now extending into the commercial setting, where when I'm talking to a patient about gene therapy, I can draw from all of that history and all of that knowledge on safety and efficacy, and really help them reach a shared decision-making point about whether they want to consider gene therapy or not. This is not like iPhones. You're not going to get a new gene therapy vector, every year. And so patients who are coming and asking about gene therapy, if there's a concern about, if you like, technology regret, well, what if I choose this gene therapy now, is next year there going to be something that works better? And I think the harsh reality there is absolutely not. If there was a better gene therapy system that was coming for patients, we would already know about it now. We would have seen the preclinical data. We would know that the clinical trial programs were advancing, and we would have some vision of when we would expect some sort of regulatory approval. So we've mentioned some of the active ongoing clinical trial programs, but you're talking about a 5 to 10 year window to take something from that's even well established in preclinical models, moving it to a clinical trial, and then getting to regulatory approval. And if it's in clinical trials now, you still want to have adequate follow-up so that you can have a conversation with patients about what the safety and efficacy of that intervention is. So I think the harsh reality for those who are considering gene therapy right now is what we have now is what we have to offer for gene therapy. And if they're interested in a brand new technology and want to get involved in a clinical trial that's ongoing, I think those availabilities will still be there. But there aren't going to be, if you like, incremental improvements in the current platform that patients should be waiting on because it's coming next year or the year after. That's just not going to be the case.
Yeah, so I totally agree. I mean, I think we have a very good factor 9 gene therapy product, which is durable and producing therapeutically very relevant levels of factor 9. I think the next gene therapy product that enters into the clinic is now some years away. So the good news is there's still a lot of effort taking place in academic institutions and in companies to look for further innovations. And those will occur at a rate that's at least as good as it's been in the past. But converting that into a clinically applicable product is going to take time. So I think we shouldn't give false hope to patients who say, well, I'm going to wait a year or two and see what's there. Nothing much is going to change in that time. You're going to have to be patient.
Now there is some great news along the way, and that is we've seen continued innovation in prophylactic options for patients that are really doing a great job in reducing the burden of how we do prophylaxis. They're improving outcomes for bleed control, patient-reported outcomes that are improved. We have shifted now from heavy reliance on IV therapeutics for prophylaxis to now subcutaneous-based prophylactic therapies. We've had 3 approvals of new therapies for hemophilia just in the past 12 months or so. And these are working their way into the clinics. And what that means is these are not definitive curative therapies, but they're highly effective prophylactic therapies that are providing a great opportunity to allow patients to live their lives until there is a definitive therapy sometime in the future that is going to be attractive to them. Now, in some ways, I would say that I think we're already there with the currently available Hem B AV gene therapy. There are some things where you have to go in with eyes open wide about, maybe the possibility of having to go on a course of corticosteroids for a period of time. But when you look at the outcomes from the Hemgenix clinical trial program, I think it's quite impressive. I think we have room to improve, certainly with Hem gene therapy. But when I look at the innovations that have come to the Hem space for prophylaxis, it's really exciting. And there's more coming. New non-factor therapies that promise higher potency, better bleed control, and maybe even taking patients up to levels that are allowing them to be bleed free on an annual basis. And so I think patients still want to have a curative therapy some point in the future, but these other innovations are continuing to raise the bar. And so whatever that gene therapy innovation that really delivers, it's now going to be compared against a completely different environment that when this current gene therapy programs started. They were comparing against traditional standard half-life prophylactic therapies. And now these new therapies are being compared to very different types of non-factor therapies. And so this is going to really ensure that these innovations they really have to deliver to encourage patients that this is the next wave of the future.
Totally agree. Of course, I think a subcutaneous injection once every 4 to 6 weeks is not a major treatment burden. So I think the only way that you would say that gene therapies are going to be significantly different is that it would offer the possibility of this hemophilia free mind, where you receive a treatment, and then for the next several years, maybe many years, you don't think about your hemophilia because it's fixed, which the current and some of the future prophylactic therapies you've been alluding to are amazing. They're very effective, very safe, but they still require attention once every few weeks with a needle. And so, well, actually, we should also say, of course, there are potential pills coming as well. So I think that the levels of hemophilia innovation continue to be incredible, exciting, and great for us to be thinking about and for patients to be thinking about too. One thing that I hesitate to say this, but there's still the issue of funding. These things are expensive, and we're talking about gene therapy in North America, so a very developed area of the world. Do you think that this will ever be something that could be internationally applicable? I remember gene therapy meetings I went to many years ago now, where they said, well, this should be like a vaccination. You give the vaccination and the patients are fine. But I think at the moment, the financial challenges are very difficult, right? I can't imagine this being.
No, we're seeing that in the US as well. I mean, I know our health systems are different, but they still have the same financial constraints. Just who's responsible is the difference. And I've seen criteria in place with insurance companies where basically they've raised the bar so high they're not even they're being transparent. We don't want to pay for this. And it's really made, in some cases, this therapy inaccessible for individual patients. I think there's no possibility of a multi-million-dollar therapy having real inroads in 80% of the world, which can barely afford to manage acute bleeding, let alone that kind of a definitive prophylactic therapy. So I think we still need to be thinking about lower tech innovations. We certainly want to have the durability in the developing world, because it's even more critical for them to be able to take a young patient identified with hemophilia and change their trajectory of bleeding risk as early in life as possible, so that they don't need to be consuming clotting factors or even non-factor therapies, and those could then be repurposed to another patient. I think that's a critical potential solution for the developing world. Identify a patient early on, give them a one-time treatment, and change them from a severe phenotype to hopefully at least a mild phenotype, if not normal. And can AAV deliver that? I'm not sure. Maybe it does need to be a lipid nanoparticle type solution. There is an innovation. Our colleague, Dr. Srivastava in India, has a program using a lentivirus and a low-intensity transplant to transduce hematopoietic stem cells that leads to what seems like curative expression of factor VIII. Is that really extrapolatable across the developing world? I'm not sure. You look at, because we saw this at the most recent WFH Congress, gene therapy using traditional AAV gene addition being developed in China at markedly reduced cost compared to other therapies. However, even if, let's say, it costs $50,000 for a single treatment, that looks great in a North American setting. However, in China, $50,000 per patient is still not doable. At least that's my understanding of the economics there. So I just don't know if gene therapy is going to be able to deliver in the developing world like the patients probably deserve and like we would like to see happen. But at least as a vision, I think we need to keep striving for some sort of a low-tech curative and intervention.
We're getting close to wrapping up now. So I'm just going to say a couple of words and hand it back to you, Steve. I tell patients that I see in the clinic now, who generally are doing very well because of all these other innovations that they're using, the prophylactic treatments. I tell them the reason they need to come back and see me is so they can tell them what is going to be coming in 1 or 2 or 3 years' time. Because if they don't keep coming to the hemophilia clinic, they will miss out on some of these innovations which will make their treatment burden less and make their quality of life better. So it's been an incredible time for us to be involved in hemophilia care over the past 2 or 3 decades. And I think it's very encouraging that this innovation will continue. So it's been great to be with you. I'll hand you back to Steve to finish off here.
Yeah, no, this was fun, David, to reflect on the journey we've all been on in gene therapy for hemophilia. I think the estimates of patients who've received gene therapy across the world is above 500 now. And I would say the great majority of those patients are living their lives not having to think about their hemophilia. And so we think about the journey we set out on in the early 90s and all the things we learned along the way. I think in many cases, the investigators, the pharmaceutical companies, partnership with patients in the centers, they've really been able to deliver a platform of therapy that it is exciting. It's definitely changed people's lives, but it's also highlighted that we still have some work to do. And hopefully, if we have another conversation like this in 5 years, as you said, we'll be talking about maybe different things, but maybe even more successes and things to get excited about for the future. So thanks for joining me today. And thanks to all of you for participating. Thanks again for ISTH for allowing us to do this webinar, and for our sponsors today for supporting this activity. And hope everyone has a great day.